Hi, Mr. Pope.
Hello, Brandon. You know there can't be. We've talked about how there can't be two of us, right?
Yeah.
So what?
So you
What?
have to leave, right?
No. You know what has? You know what has to happen, right?
What has happened?
One of us has to take the other one out.
All right. Our
Two go
focus
in.
goal.
Two go in. One come out. One comes out. I haven't been beaten in a long time, dude. Can't be beat. What's up, Cam? Two goes in. One comes out. What does that mean, Cam?
Two goes in, one comes out.
That's right. What does that mean?
It's not interface, is it?
Interface. Oh no, I'm not talking about any kind of science stuff. I'm just talking about
Two goes in.
just two goes in and one comes out. What does that mean?
I don't know. Two goes in, one comes out.
Yes, sir.
Oh no.
Two go in.
What's the answer?
Well, ask Brandon. Brandon, what does it mean?
What does what mean?
Two goes in, one comes out. What does that mean?
Isn't that like the octagon, like the UFC or whatever?
That's right. That's UFC, baby. That's UFC.
Oh.
That means I go. That means Brandon and I go in, and only one of us coming out. It's not. It's not going to be Brandon. Oh, Samba. It's going to be Pope. I don't know. Couldn't tell you. Couldn't tell you. I don't know. Time's class start 12:05. That's what time we're going to start. Make predictions. Who thinks that Pope's going to win over Osamba? Who's going to win? Osamba or me? Uh-oh, I get some guesses already. We're getting guesses. Earl, you're playing it down the middle. You're playing it safe. You got to take chances. You got to take chances. You got to take risks. You got to take risks. You got to take chances. You got to pick a side. Can't stay in the middle. Can't stay in the middle. Let's see. Oh, good Lord, Tabitha, you're screwing up my chat, man. Oh, okay, gotcha. All right, we'll leave it. We'll leave it be. We'll leave it be. We'll leave it be. Good morning. Most of you guys have apparently been able to find the workaround to get onto Schoology. I'm curious, how many of you guys are actually on Schoology? I know that there was an issue this morning. How many of you, just drop a Y, I'm on Schoology, just a Y for yes in the chat box. I am on Schoology. Awesome, awesome, awesome. Excellent, excellent. Okay, everybody's got that fixed then. I think the lesson here is to bookmark it. If you are still locked out, once you get in, bookmark it so you don't have to go through ClassLink. That'll probably make your life a lot easier. I'm going to take an answer to this question at 12:05. So go ahead and start working on the do now. Man, is it just you and I, Cam? Oh man, they can't find out what goes on in here, man. Oh no, we don't want any witnesses. I don't want any witnesses. I don't want any witnesses. It's just us. I was telling him it's just going to be Cam and I, and then there wouldn't be any witnesses. It'd just be hearsay. It'd be his, you know, what he has to say against what I have to say. And I don't know. Like, he did this. I say, no, he did that. Who do you believe at that point? You know, nobody. Just even. You just have to keep our mouths closed. Again, we'll take an answer to this question at 12:05. You probably, if you guys are on the computer, probably see it better if you maximize your screen to make it a little larger so that you can see it. I was told you have a nickname, Brandon, by the way. Several people said you have a nickname. I don't know what that is. But they said you would stay inside, you wouldn't leave. So 2 minutes. I'll come to one of you guys. Don't text me your answer yet. Some of you guys are already sending me your answer. Let me make sure you guys can share your screen when we go to breakout rooms. You should be able to share your screen, yes. Woo. I got a new toy today. Oh, no, I didn't mean to do that. Sorry. I got a new toy today. Let's see if they can get this thing to work. There we go. Excellent. 1 minute. Man, where is everybody? No, don't start sending me your answers yet. Don't send me your answers yet. Do not send me your answers yet. All right, on the count of, I'm gonna count down from 10, and everybody send me your answer on the count of 10. You can send it to me privately, or you can send it to me in an open fashion where everybody can see it. Doesn't matter. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Go. Drop your answer. Drop your answer in the chat box. Okay. It's A through D. It's not a yes or no question. A through D. So far, I've had 3 correct answers, 4 correct answers, 5, 6, 7, 8. Okay, so people are changing their answers based on which one I said. All right, so you are correct if you answered D. And let's look at why. Some things about this question that I would have left off. I would not have said a thing. I would not even not said, but I would not have, let me let Aspen in here really quick. Some of this information was not important at all. There was nothing important about this information up here. I did not need to read this. And I know that because the question is asking me about the data. So when I want to look at the data and see what the data says, I'm going to come up here and I'm going to start looking at these numbers. Now, I'm not even really that worried about what these phenotypes are. I just want to start looking at my ratio. So I will bring in the phenotypes, and I just want to see how often is gray and long together versus gray and Apteris. It doesn't even matter what Apteris means. Apteris does mean vestigial wing, or like little bitty shriveled up wings. So I'm going to just do 42 divided by 9, and then 41 divided by 8. That's going to be, we'll put a little star here. We're comparing these two. What are my numbers for these? 42 divided by 9. If you have a phone, you can do it. So I should see people in here doing it using their phone. 42 divided by 9, 42 divided by 9, 41 divided by 8. I'll call on you guys in just a moment. I'm only going to spend 5 minutes on this due now. I've got to keep a schedule. So it's 12:07. I'd like to be done with this by 12:09. So let's go to Cam Boyce for the first one. 42 divided by 9. What is that number, Cam?
4.67.
I'm sorry?
4.67.
4.6. Oh, crud, I can't write over here.
It's like a bunch of sixes.
4.6. Okay, so 4.67. So actually, let me get my eraser and get that off. And let's make it maybe write a little smaller. So that's going to be 4.67. So would I round up or down? I'd round up. Okay, excellent. So 41 divided by 8. Let's go to Brandon Only 2 go in, 1 come out. Let's see if Brandon comes out. 41 divided by 8.
Do you want me to answer?
Yeah, I want you to answer it, man. Do you want? You're not gonna send it to me.
Can you repeat the question? I'm sorry.
41 divided by 8, dude. Right? Oh man, look. I'm blaming you for that. All right. 41
It's
divided by
5.125.
okay, 5.125. Do we round down or round up? 5.1 or 5.2?
Down.
Down, you're right. Okay, so essentially that's what I've got. If I had to put this into a ratio, let's just cancel these out and make that 1. And then I'm going to go ahead and put this one 4.7, and this one is going to be 5.1. Essentially, my ratio is going to be 5 to 1. So if that's true, I am not going, none of this, all this up here was irrelevant, just like on the ACT. You read the question if you have, or read the prompt if you have to. Look at what the question's asking. They're asking you about the data. Here's my data. The rule that I was telling you before, if I was doing this and we knew these were heterozygous double het, we would have a ratio of like this. You can answer this question, and the retake is Friday. This is one of the most infrequently correctly answered questions that we had. It's all about if they are linked. Now, I have gone over this twice in office hours. Not this particular question, but a question like this, and done a lot more math than this, and even gone further in with chi-square. If you haven't come to office hours, you want to do the retake, you need to come to office hours tomorrow. But I would watch the video from previous office hours, which I've attached to the same link for your test. Go back and watch that. So those of you guys that got D, you got it right. These genes are linked. They are located closely together on the same chromosome. That is it. So not a hard question to get, not a hard question to answer, but one that you're going to have to look at over and over and over to be able to do it. All right, everybody should go to Schoology to go to today's lesson. Thank you for participating. We are on schedule. We are exactly on schedule. Let's go to messages. Let's go to today. And I'm going to go to class content. Our unit is still genetics. We just broke this unit into parts. So we had 2 summatives. Let me give you your other summative today. Today, we're going to go through facts and rules. Now, before I get, this will be the folder we're working out of. But before I do it, I want to go back. I want to look at our agenda. What are we doing today? So for facts and rules, there's a few things that we have to do. We've already handled the do now. That is over. Now we're going to be working on the INM. I am going to have the INM, and I promise not to go more than, in this case, well, my little marker doesn't work on this thing very easily. I promise not to go more than 15 minutes. If I do go more than 15 minutes, then Cam gets to tell me to shut up. So you're going to set a timer when I say go. Set a timer, 15 minutes. You tell me to hush or shut up or something. The guided practice I have here for you guys is 20 minutes. I'm going to try to avoid that. I'm going to try to avoid spending that much time on the guided practice. I think you guys will probably get that done in 20 minutes. I really do think that you guys, get that done in 15 minutes. The last class got it almost completely done in 15 minutes. So we're going to shoot for that. The exit ticket, I'm going to save for the asynchronous retake. There's instructions for the asynchronous day. I'm going to save this exit ticket for that part. This is the easiest grade you will ever get. I will shift that document down there. I'd rather spend some time right here on this individual practice, this IP, because this is going to be your next summative. So we'll carve out a little more time for here and carve out a little time for here. At least that's how it worked out with the last class, and it went really well. So I'm going to get into it. I have attached the notes. I don't want you to look ahead of the notes. I really want you to focus on what I'm showing you, because I'm going to show you a couple of little tricks today that you can use to answer questions when we get to, get a little further along into this unit. So first things first, we'll leave off where we left off last time. And last time we really wanted to know, how did they know what it was, DNA or proteins? We answered that question. So what about now? So now we know that people have accepted that this is the code. This time we said that they did finally accept it, that DNA is the code for heredity. So let's look at what the structure is like. So I really want you guys to look for patterns. We're going to be pattern seeking today, looking for patterns. So I'm going to pause here 30 minutes. And again, if you are not making your screen full screen, you need to make your screen full screen. Make your screen full screen so you can see this diagram, this image. Thank you. And I'll wait 30 seconds. Do not put your answers in the chat. Not yet. I want you just to look. We're going to start at 15 minutes if you don't mind. 10 seconds. So I see some of you guys are putting it in the chat. I'm not going to look at the chat until the time is up. I know you're seeing patterns. There's two primary patterns that I'm really looking for. So if you've already found one, see if you can find the other. Okay, drop your patterns into the chat. Drop your patterns into the chat. I don't know what that means, Alosa. Could you come on your mic and tell me? Okay, you see some shapes that are patterns. Okay, I see that. There should be a little more. There's some elements. Okay, you got one of them here. I still looking for others. I see what you're saying, Jalen. That's not enough data to get a pattern yet. Yes. Tabitha, you essentially what you're saying there is that you're saying the same thing twice. I'll come back and mention that. Still one more pattern. Nope, don't worry about that. Yep, those are previous units. I see that. I can tell what it is. I see that. Still looking, there's another pattern. So let me establish one pattern that's already been noted. So let's see. One person, and Tabitha said that a base, these bases are always connected to the sugars. A base, a nitrogenous base, is always connected to a sugar. What is the other pattern that you see? There should be another pattern you see. So some of you guys are telling me the pattern of TACG. That doesn't repeat itself, so that can't be a pattern. A pattern is something that occurs over and over and over. So my bases I have attached 4 times. It's happened here, it's happened here, it's happened here, and it's happened here. The bases are always connecting to the sugars. I see that happening. Where else do you see a repeating? Good, thank you. So yes, the phosphates are also connecting to the sugars. This is always the same. This is a repeating pattern. These molecules, the phosphate groups, are always connected to the sugar. There's no change. Therefore, the code cannot be here. The code is going to be in this here because we don't have enough data to see if there's any kind of repeating pattern here. The only pattern we see is that the nitrogenous bases are connecting. That's the only thing that we can see so far. So that's all I really want to get. Let me see if I can get this off. I've got a new, I hope you guys like my new toy. I got a new toy and I am trying to use it effectively. Yeah. And I'm just practicing with it today. It's going to make it a little easier for me to draw here for you guys and not use a mouse. So got it this morning from Amazon. All right, let's try the next slide. So we are going to look for another pattern seeking. There's a person named Chargoff. And the difference in IB and AP didn't give a flip-flop if you knew who this person was. IB wants to know who this person is and what they did and what they noticed. So that is going to be important. You do need to know Chargoff and his rule. Here's his rule. I want you to see if you can find a pattern here. Don't say anything to anybody yet. Just look at the data and see if you can find a pattern. This is a very rule-heavy, fact-based unit. You probably have noticed a pattern by now. In the chat box, I need you to drop the answer into the chat. In the salmon, what is the percentage of thymine? What is the percentage of thymine? I should have 25 answers. I have 3. If I don't see 25 answers without even looking at the chat, I will call on one person. What is the percentage of thymine in the salmon? Okay, let me get a picture of that. Thank you, thank you, thank you. Those of you guys that took the risk, you all get 1 point on your next quiz for following instructions. Congratulations. Thank you for doing that on time. All right, so yeah, you guys are seeing it. Now a lot of you guys decide to participate. Maybe we'll get more next time. So here I have this 29.1%. So let's try this again. In humans, what are the percentages of guanine and cytosine? I'll wait a full minute for that one. I'll wait a full minute for that one. What are the I'm sorry? What are it's right there on the what in humans, what are the percentages for guanine and cytosine? You're looking at salmon and asking about humans.
Oh, yeah.
For guanine and cytosine. Nobody's got it yet. Nobody has it yet. Not looking for salmon, I'm looking for humans. Nobody has it yet. You can give me an approximate value as well. I'll even take an approximate value. You guys are guessing without looking at the pattern. All right, let's solve it. Let me solve it for you. Oh, Earl, you got it right at the last minute. Earl found it at the last minute. So let's look and let's see. Anytime we have DNA, I've got 4 parts of DNA. All 4 parts are going to make a whole. So if I add all of these parts together, that's going to equal 100%. So in humans, I already see 30.4 and 30.1. So let's see, 30.4, 30.1. That is going to be 60.5. Well, I'm looking for 100. The total over here is going to be 100. Every time I add these, they're all going to equal 100. So 100 minus 60.5 is going to equal 39.5. Now, these, if you'll notice, these tend to be relatively similar, equal numbers. You can notice that adenine is the same as thymine, almost the same, not exactly, but very, very close. So there's a pattern emerging. So we were trying to figure whatever's in here should be, if that's 30 and 30, this should be close to 20. Should be close to 20. Let's find out. 39.5 divided by 2. What is my number? 9. Somebody put it in the chat box. Let's see what we get. 19.75. You are correct, people. You are correct, people. So 19 point. What is happening to my stupid thing? It's going to make me hurt it. Come back over here. 19.75. So that's close to 20. So if you were being asked a question about this, I would just look for whichever number is closest to 20 and move on. There is a pattern. You'll notice the pattern is pretty much the same. All of these are close to 20. All of these are close to 20. These are close to 20. This is my exception right here. I put a little dot. So these are all close to 25. So you could assume probably 25 and 25 here as well. They should match. So this is the Chargaff's rule that says guanine is going to be equal to cytosine and adenine is equal to thymine. And you guys may remember that when you're a freshman. It's a rule that you're going to have to use for DNA. For DNA, we'll have to use that. So how did we get to these answers? Again, we didn't know what the structure of DNA was like. It was very hard to know. And it took a woman to figure these things out. Women tend to be very excellent scientists. So please take a look, especially if you ladies are interested in biology at all. One of the best scientists in the United States today that's very young, in her 20s. Her name is Martha Munoz, and she is a boss. I'm an avid follower of hers. I love that and admire her a lot. She's done amazing work. But Rosalind Franklin is the person who developed the idea that the DNA's bases were inward. Here, Watson and Crick, they figured out that it was a double helix, but in terms of the bases being inside the helix, that is going to have been Rosalind Franklin who used X-rays. That is actually something you have to know. You have to know these things. And I'm going to insist on it. By the way, do you guys see any patterns here? Do you see Chargaff's rule in this example? Yes, you do. You can see it. So you can see guanine equals cytosine, thymine equals adenine, thymine equals adenine, guanine equals cytosine, cytosine equals, and you can see it over and over and over. So it was Rosalind who figured out that it was an anti-parallel, and what we call an anti-parallel double phosphate backbone. And I'll show you what I mean by that. It's right here. Now here it's anti-parallel, meaning it's going two different ways. So you'll notice that there is what we call a 3-end on this molecule. This thing is driving me nuts. I'm practicing with a new little pad that I bought for this class, that you have a 3-end and you have a 5-end. You have a 3-end over here. They're on other sides. So again, 3 is here, 3 is over here. This is an anti-parallel. They're going both directions. You'll notice that these sugars, pentose sugars, the point is down. It's always facing the 5-end. It's always facing the 5-end. It's going up here. It's always facing the 5-end. And that can kind of help you answer some questions as well. Now, hydrogen bonds. How many hydrogen bonds are between? Actually, let's just, I kind of went ahead and looked for I should have asked you about a pattern, but how many hydrogen bonds between thymine and adenine? How many hydrogen bonds between thymine and adenine? Good job. Good job. There are some people, I got 2 different answers, and I knew I would. I got 6 and I got 2. And the answer is 2. These little dots are all symbolizing 1 hydrogen bond. These little dots are broken. And do you guys remember why? And guanine and cytosine have 3. Do you remember why the dots are broken? Are hydrogen bonds, are they weak? Are hydrogen bonds weak or are they strong? So we'll put weak or strong. Which is it? Which is it? Which is it? Weak or strong?
Weak.
They are definitely weak. They are weak, just like Cam sitting here. He's got the weakness. I'm sorry, Cam, what'd you say? Brandon's weak. Oh, yeah, Brandon's weak. No, Earl's weak. Now he's saying Earl's weak. Yes, these are weak like Earl. Very weak bonds. They're easily to break. You can break these bonds. And this is one of the reasons that in Griffith's experiment, that even when you heat, even when you add heat is going to break. Whenever he heated those bacteria, it did break down. That heat did break or denature, and I'll put break, denature the proteins. It did do that to proteins, but it did not do that for that heat in Griffith's experiment did do that, but not DNA. DNA has rules. And the thing about it is guanine and cytosine have 3 hydrogen bonds. So even when they're broken, they're going to go right back together. They have these attractions that allow them to go back together, just like water. If you let water go into the atmosphere and then it cools and it condenses, the hydrogen bonds reform. So in the heat did not, in this case, did not, in Griffith's experiment, break down. So Griffith's experiment with the bacteria. Is that my 15 minutes? What are you going to tell me?
Shut up.
Shut up. He told me to shut up. He said, shut up, you got weakness. And I said, no, that's Earl. This will be the last thing I bring up. So Griffith's experiment, get this thing out of the way. And Griffith's experiment did not break this down. Did not, these were able to reassemble. These could reassemble. I'm going to get that back over here. These could reassemble. The DNA could come back together with no problem at all. And therefore it transformed the bacteria. All right, I'm going to release you guys for a moment. Before I do, I'm going to go to the website. I've got through most of what I wanted to get through today, but I wanted to stay firm with my 15 minutes. So here you've got some things that you are going to use. First thing we're going to use today is our guided practice. So I'm going to open this up. When I open this up, you're going to see a ton of questions. You're going to answer as many of these questions as you can in 15 minutes. As many of these as you can in 15 minutes. You will not be able to answer all of them. You will be able to answer probably about 75% of them. Most of these we have already learned either in this class or the last class. When I come into your breakout rooms, what should I expect? Screen sharing. Screen shares. And what was the other one, Alan?
Talking.
Talking. And talking in a very monotone voice like Alan. Exactly. I'm kidding, Alan. All right, so you guys get ready. I'm going to break. You guys should be able to get in there and see how many of these you can get done in 15 minutes. I bet you can get 75% of it. I love giving you guys a hard time. It's the best part of my day. So you're weak. Weak like Earl. Where's Earl's group?
All right, guys, open up the document.
Open the document. Yes, sir.
Brandon, open the document, Brandon.
Okay, can you screen share Earl? Or?
So Tracy, Aspen, and Emma. What's up with you guys? All right,
The other one is nitrogen. Oh, never mind.
No tubs. Hang on.
Yes, DNA and protein. Damn. The first one is DNA. Oh, no, never mind. Never mind. Okay, so it says, for number one, it's a 5-carbon sugar. What?
A lot of absences today.
For number three is phosphate. And then the second one is OH.
Which one?
Number 8, the last one.
Yeah, they're not transmitted. That's right. They were not transmitted. Who was that said that? That was Muhtar?
Donna.
Muhtar, did I just count you absent? I think I did. Oh, no. Hold on a second. I gotta leave this room. I gotta go change attendance. Hold on, I'll be back.
James Watson. Is it Helix?
Great.
A double helix.
A double helix. See. Double helix. These are just things you have to just know. I want to make sure you guys don't miss like the easy questions. Knowledge base.
Thymine is complementary to adenine.
Who are the people speaking?
Me and Jessica.
Say.
The first one I think is 2 for 13 is the second process. And then 3. Yeah.
Some of these you're not going to know just yet. You don't have to do all of them, but you can look at them. There's going to be some of these that you just don't know yet. But it's good to kind of see what's coming down the pike and you'll be like, oh, I remember that. Those are things I have to look out for. All right, I'm going to go check out the other groups.
Okay.
Repeat the question.
Oh, shoot. There's somebody that's locked out. I got to.
Mr. Pope, sorry, it's just my internet started acting up. That's why I couldn't get into Zoom. So I'm using my personal hotspot.
Don't worry about it, baby. Which room were you in?
Huh?
Which room were you in?
I was in room 2 with Romello and Daisy, and yeah.
See.
To ask about that one.
Ask about what?
Oh, question 8. We don't know.
So DNA, I'm gonna just check the other one. So DNA was labeled radioactive phosphorus. Yes. Protein was not labeled. Oh no, it was labeled. What was labeled on the what they put on the DNA?
Sulfur, right?
Simone. It was labeled. So that's false. You may even want to put out to the side. It was labeled with sulfur. Radioactive sulfur was found in the offsprings. Okay, so was it found inside the offspring phages or not?
I don't think so, because wasn't it DNA that they found, not protein?
Exactly. You're correct, ma'am. You're correct, ma'am.
So would that mean the last one's correct then?
Phage proteins were not transmitted. That is true. They were not transmitted to the host cell. Excellent. You guys are thinking. I love it. Y'all are my favorite class. Love this class.
You like us even more than 2A?
You mean 1A?
I mean 1A? Sorry, 1A.
Yeah, because the 1A, I'll be honest, 1A usually outperforms you guys, but they are sticks in the mud. They don't talk, they don't hang out, they get all head in the oven sand. You guys are just like charge, fearless. I love it. So much more fun. Tell
I'll
them I
make sure to let them know.
Tell them.
Species contains equal amounts of A and P, right? Adenosine. Is it adenosine? Adenine? What is it?
Adenosine? I have no idea.
Adenine.
Oh.
You can say adenine or adenine, it's not gonna matter. If you're from the Northeast, you'll say adenine, or if you're from the South, it's adenine. You know adenine.
Okay.
Thymine,
That's why.
Chargas rule.
Thymine, and then the one that starts with the G, and then cytosine.
Guanine. Guanine.
Thank you.
Welcome, baby. Grace is your cousin, right?
Yes, sir.
I talked to her about 30 minutes ago.
Oh, really? Dang, she couldn't even text me back, but
She was on the phone with me.
red.
All right. I'll see you guys. My alarm's about to go off. I gotta close the rooms.
Okay.
Roger, Dodger. I'm waiting for everybody. Some of the groups are still working. Group one, nobody left the room. They're still hanging in there. They're like, we're gonna get as much done as we can. Oh, want to be there forever. Excellent. Now let's go back and see what else we have to do today. Now, a couple of reasons that I gave you guys these questions. One is, do you know a lot more about this than you realize? Two, you guys discuss it. If you discuss it, you remember it. If you remember it, you do better. Third, I want you to see what kinds of questions I have coming up. Like, what should I be looking for? Kind of have an idea of what am I going to need to, what's important. So you saw, so there were several questions like, I don't know what the heck that means. You're right, you don't, and you won't until next week. But at least when you see it, you'll know it's important. So you should keep this document handy. Next, we got to go to our quarter project. This one is a summative this time. So let's, and it is going to be individual, but people can work in groups to help each other, but everybody will have their own. So first thing I'm going to do is I'm going to ask you guys to go to the DNA model rubric, open it, and then you're going to have 5 minutes. I'm not going to say a word. You have 5 minutes to look through it and ask me questions about the rubric. 5 minutes. Oh my God. Who put the Bernie Sanders picture in the hall? Y'all know?
I thought he was one of the people in the staff.
Really? I think it's hilarious. I love it.
I thought it was real when I walked in.
Oh my God, if Bernie Sanders is downstairs, I'd trip out. It does look real. Look. So funny. Let's go. 3 more minutes. Read through the rubric carefully. Be thinking, what do I want to ask Mr. Pope about? After looking through this very thoroughly. What? All right, we're getting very close. Please don't chat your questions to me, please. All right, so please, if you have questions, please don't chat them. Please ask them out loud. Everybody will hear them. I'll hear them. If you don't, if your mic doesn't work, put it in the chat. One of you guys read the chat to me. Make sure.
Could we use stickers for the collage?
Maybe. The thing is that what you're going to have to do, since it's going to be repeating patterns, like you're going to have the same thing represent guanine every time, the same thing represent a phosphate group every time. You can't have different images representing the same base or the same or the sugar or the phosphate. So, or if you have this something, you got to have represent your hydrogen bonds. If you use stickers, you better have a bunch of them. But I guess stickers would work, yeah, if you've got enough of the same one to use it over and over and over.
Okay, thank you.
It's going to have to be some, and then you'd have to have a picture on it, and then take a picture, and then you could put that picture on a slide. You're going to have to present it if you're at home. The kids that are here, they have to present a physical model. They can look around the room and see those. You're going to actually have to make a structure.
So if we're online, we're basically going to be doing something like virtual, not like a structure
Yep.
or like something like hand, like 3D, like they are. We have to do it online.
You're correct, ma'am.
Can you show us an example of like past projects?
Oh my God, they're all over my room. Yeah. Hold on. So remember, everything that you have to use has to be garbage, has to be refuse, right here. See, nothing can be bought. Nothing can be bought except for paint and adhesive. So this is an example of one. You have to look at me. I'm like a jealous boyfriend. I want all your attention. You know, those guys that are really insecure and they got to have everybody's attention all the time. That's me. So this is an example. And here they use like the stripping that you would use for a door jamb. And it looked like they went to a construction site, you can tell. The phosphate groups were, they labeled with the metal here. This was the sugars, the nut, the bolt on the inside. You can see here they have them labeled adenine and cytosine. They do have labeled like 2. They got the number 2 for the hydrogen bond to adenine, thymine, 3. They got to do, yeah, this one, I think it's pretty good. And it has to be able to hand out a lot of stress. So like if I just threw it down on the ground and it didn't break. So I'm going to throw them on the ground. If it breaks when I throw it, you lose points. For those kids that are in class. Huh?
Could you give us an example of like one of the virtual ones? So
I can send you guys some. It's going to have to be pretty creative on your part. If you Google DNA collage, virtual collage, you can find some examples that way too. But yeah, I'll look for some tonight and send you put a picture up here.
Thank you.
Okay.
You said the kids that are at home can do 3D models of it.
Do what again?
That the kids that are.
Oh no! If you're at home, you need to make a virtual one. I wouldn't be able to see it. I mean, you'd sit there. How would we? You're going to have to present this. I mean, I don't have time to go. Okay, here, get your camera and look over here and there. It would drive me. Yeah, I wouldn't be able to actually see it. I need something you can post and I can come back and look at it later. Other questions?
What if we take like a picture of it?
I still don't think it would be near as good as I need to give it a really good once over. It's a lot harder to do, actually. I think making an example of one, making an actual one of these. I also couldn't test how tough it is. I'm going to throw the ones that are in here, like literally throw these things, like I did that one. You guys still need to look at the questions too, and we have to get out in 3 minutes. Can you guys also jump to the questions? Let me go over here to look at the last thing that we need to look at is the questions. So where are the questions? And you need to understand how this is going to work. DNA project questions. I'll project it really quick so you can just see this. And I'll give you guys some more time to ask me questions about this rubric as well. You can see there are 27 questions. I have a number generator and I'm going to ask you in class and you are going to have just 3 seconds to respond. I'm going to take a jar, shake it, and I'm going to pull out a number. And let's say I pull out 20. You have 3 seconds. I'll ask, how is prokaryotic chromosome different than a eukaryotic chromosome? And you better be able to answer that question. There are several things for this particular question that you would need to know. So these are you've got some time to get these, and you don't know which 4 questions I'm going to ask. So you need to know all of them. Now, most of these you can pretty much, you can get, like, where is DNA found in eukaryotes? You guys know that. Nucleus, where is it found in a prokaryote? You already know that. Nucleoid region. What are the rules for base pairing? You already know that. Adenine pairs with thymine, guanine, cytosine. How many hydrogen bonds? You already know a lot of these, but some of these you don't know. So these are also things that you need to know. Oh my God, keep these in your back pocket as what I'm teaching you guys over the next 3 weeks. Be adding these to your repertoire of knowledge that you have to be able to know. For this exam, IB insists that you know all kinds of little bitty details. So this will help. I've got 1 more minute for questions.
Can you post some examples of those virtual things eventually?
Yeah, I'll find one tonight and I'll post it under this same folder.
Okay, thank you.
All right. And then also make sure I'm going to add your exit ticket today is going to be here for the asynchronous test retake. I'm going to have office hours again, 1 more office hours tomorrow. You need to come to those office hours for that retake. Otherwise, it's just going to be the only thing you'll be able to do is take the you'll just be stuck with the first test, and only thing you'll be able to do is just come in and hit the exit ticket. And I'm gonna have this exit ticket. This is the easiest exit ticket in the world for the asynchronous exit retake. So office hours again tomorrow. It'll be the last day I do office hours. I'm gonna see you guys. I won't see you guys till next week, and so. In the meantime, you guys be good. Deuces.